Question number 146. Breeds of cattle and chicken are respectively. And the options are, very first option given is Brown Swiss and Marwari. And this is an incorrect option. We'll write here cattle, chicken, this Brown Swiss, but Marwari is an Indian sheep. So this is an incorrect option. Although Brown Swiss is a cattle. The next option is Jersey and Leghorn, and this is a correct answer to our question because Jersey is an exotic breed of cattle, whereas Leghorn or White Leghorn is known as a layer. It is a breed of chicken. The next option given is Rhode Island Red and Ayrshire, and this is an incorrect option because Rhode Island Red is American breed of poultry, whereas Ayrshire is an exotic breed of cattle. The next option is Hisardale and Mule, and this is an incorrect option. Hisardale is a cross breed of sheep, whereas Mule is a result of interspecific hybridization. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 147. The integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. The above definition of biotechnology was given by, and the options are Genetic Engineering Approval Committee, European Federation of Biotechnology, US Patent and Trademark Office, International Union of Biochemistry. And the correct answer to this question is option number 2, European Federation of Biotechnology. This body defines biotechnology and encompasses both traditional and modern view of biotechnology. Let's discuss rest of the options. The very first option given is Genetic Engineering Approval Committee, and this is an incorrect option because this committee make decisions regarding the validity to genetically modified research and safety. The next option is US Patent and Trademark Office, and this is an incorrect option because US Patent and Trademark Office gives patent rights. The last option given is International Union of Biochemistry, and this is an incorrect option because it is an international organization concerned with biochemistry and molecular biology. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 148. The linking of antibiotic resistance gene with the plasmid vector became possible through the help of, the options are restriction endonuclease, DNA ligase, DNA polymerase, exonuclease. And the correct answer to this question is option number 2, DNA ligase, is commonly known as molecular glue. And helps in linking of antibiotic resistance gene with plasmid vector. Let's discuss rest of the options. The very first option given is restriction endonuclease, and this is an incorrect option because restriction endonuclease serves as molecular scissors or chemical knives to cut genes or DNA into defined fragments. The next option is DNA polymerase, and this is an incorrect option because DNA polymerase synthesizes new strand of DNA complementary to an existing DNA template in 5' to 3' direction. And the last option is exonuclease, and this is an incorrect option because exonuclease can digest DNA in non-specific manner. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 149. The antibiotic resistance gene isolated by Cohen and Boyer was inserted in plasmid native to which organism? And the options are Salmonella typhimurium, Bacillus amyloliquefaciens, Escherichia coli, and Bacillus thuringiensis. And the correct answer to this question is option number 1, Salmonella typhimurium. The first antibiotic resistance gene isolated by Cohen and Boyer was inserted in plasmid of Salmonella typhimurium. Let's discuss rest of the options. The next option given is Bacillus amyloliquefaciens, and this is an incorrect option. This bacterium belongs to the genus Bacillus, which is a source of BAM H1 restriction endonuclease. The next option given is Escherichia coli, and this is an incorrect option. Escherichia coli is a bacterium closely related to Salmonella typhimurium, and it acts as host for cloning of antibiotic resistance gene. The next option is Bacillus thuringiensis, and this is an incorrect option. Bacillus thuringiensis is a bacterium which infects plants, and this bacterium contains insecticide endotoxins. So the correct answer to this question is option number 1. Let's discuss our next question. Question number 150. Plasmid DNA cannot. And what are plasmid DNA? Plasmid DNA are extrachromosomal, autonomously replicating, closed circular DNA. So they cannot be double-stranded. This is an incorrect option because they are double-stranded. They cannot be coated with histone proteins. And as it is a correct option, so this is a correct answer to our question, because chromosomal DNA is coated with histone proteins and not plasmid DNA. Plasmid DNA is naked and it lacks histone proteins. The next option is plasmid DNA cannot replicate independent of chromosomal DNA, and this is an incorrect option because plasmid DNA are autonomously replicating DNA. The last option is circular in shape, and this is an incorrect option because plasmid DNA are closed circular in shape. So the correct answer to this question is option number 2. Let's proceed to the next question.